They what? Sorry.
And I took a walk through the area and at the University of Tasmania Medical Campus in Sydney.
Oh, really?
It was kind of weird.
Yeah, yeah. Universities kind of metastasize everywhere these days, nationally as well as internationally. Monash has got branches in near Florence and in Malaysia and China, and yeah, they're global businesses these days.
Is that for business reasons or research reasons or?
I don't know, but their ranking's going up, so it's not hurting them. So
yeah, but how do they get their ranking?
It's like a whole.
Go there.
It's a whole of the movie. So it's a whole. It's a whole of the doco.
So you're in research. How did you end up there? How did you start? Was there any?
Yeah,
You can think about that for a sec. Was there any moment you went, I know what I want to do? Or, of course, one person actually said, I did science and then I got to university and the queue for modern history was too long, so I wanted the science.
So it's a bit of an uninspiring story, actually. The only quirk around that answer is that there's this family story that my mother tells me that when I was very young, like I don't really remember this, but apparently I was standing at the helping my grandmother do the dishes after dinner or something like that, probably getting in the way. And apparently she asked me what I wanted to do when I grew up. And apparently I said, oh, Granny, I'm going to cure cancer. And I didn't know about that, but my mum, as I've become increasingly involved in research in the last sort of 20 or 30 years of my career, it's like mum sort of started telling this story about that. But I have basically no recollection of that. So for me, practically, even though it might have been predestined in some way that I wasn't aware of, I'd say it kind of crept up on me. I mean, look, I was pretty lucky. School came quite easily to me. I went through that pretty well and got into med school at Melbourne Uni. I sort of always thought that I would do medicine, although in truth I probably didn't think a lot about it going through school. I was just like kind of a regular teenage kid just doing his thing. But I did really enjoy medicine, particularly the latter years, working in hospitals and starting to interact with patients, and I really felt like this was my home. And then, actually, even in terms of getting into cancer medicine, I actually had no inclination to even become an oncologist like I've become until reasonably late in my hospital-based training. So I sort of finished med school. I was working like as a young doctor in the hospital, just running around doing things. And it was actually the last rotation before I had to really figure out what I wanted to do after that initial training period. And I had very little exposure to cancer medicine up until then. I'd done gastroenterology and cardiology and neurology, like other medical disciplines. I didn't have much exposure to cancer, but it was quite amazing. Within a week or two but my last rotation was to go and work on the cancer ward at the hospital. It was like a big general metro hospital I was training at. And it was amazing. Like within like a week or two of being on the ward, I thought, oh my goodness, like this is what I was meant to do. It just felt so natural. I had not a lot of inclination prior to that would be. So I felt almost a bit serendipitous, really. I was also lucky. I had great mentors, and it was a great team. There were excellent supervisors and good leaders of that unit at that time. And so I think there was just a real kind of feel-good environment, albeit in an incredibly difficult area of medicine that was extremely challenging in many ways. But I mean, I was within weeks of studying there, I was hooked. I mean, I knew that this was what I was meant to do, and so I was already kind of committed to doing. I think it was neurology. Actually, I'd already sort of signed up to train as a brain specialist, but I actually pulled out of my. I sort of pulled the application and then put in an application to get into oncology training, which is what I ended up obviously doing. As far as research goes,
no, that was the next question.
Yeah, no. So
you pick oncology now. No, there's two levels here. You pick one now. We're going to research.
Correct. So and so as far as research goes, again, when I was starting oncology training, I really didn't think much of it. I mean, I found oncology is a very research orientated specialty. There's a lot of focus on clinical trials. Probably arguably the leading, or if not one of the sort of top medical subspecialties in terms of its like very rigorous discipline around clinical trials and really testing whether or not new medicines work or not. It's a very strong part of the culture. So I kind of grew up and I was trained by leaders and experts in oncology who had sort of developed and carried that culture. So I was a little bit imbued with that. But I reckon it probably wasn't until probably the third year of my training where, again, a little bit serendipitously, I happened to have some wonderful mentors and immediate supervisors that I worked closely with who were career clinician academics. And I sort of got a little bit of an inkling about what their lives were like. And I kind of liked it. I thought it was pretty interesting, the breadth and depth of stuff that they engaged. But I reckon it was probably mostly as I increasingly started to treat patients with cancer, usually, this is sort of back in like the mid-1990s, mostly with chemotherapy drugs that, although in many cases were useful and effective, really actually were often not that good. Or if they were good, only very temporarily so. And so I think I had this real sort of dawning sense that I was enthusiastic about this path that I was on career-wise.
Actually, yeah, but I wouldn't mind being writing. What path did you select? Because we know what you do, but we haven't told the audience what kind of research you got into and why.
Yeah, so I'm kind of coming to that actually. Because there's a theme that actually links back from then all the way through to where I would have ended up really focusing on myself. So one thing's very frustrating about being a doctor that treats cancer patients is that although some of our treatments work quite well initially, often reasonably quickly, the cancer becomes resistant to the treatment. And so I think increasingly as I was actually doing this and treating patients and seeing what was happening, it's actually, so it's awful for the person. It's actually awful as a treater as well, because you kind of vested in the outcomes of the people that you're looking after, and it's pretty disappointing to very frequently have these sort of failures, if you like, or patients that develop recurrent disease that you ultimately can't treat. So I got this strong sense towards the middle and end part of my oncology training that like what we actually need right now is not more and more doctors prescribing drugs that don't work very well. We actually need more doctors who are invested in research that will be the basis of the discovery of new treatments and new drugs that will improve the outcomes of patients. It became pretty obvious towards the end of my training and I felt quite engaged in and passionate about that. So it was sort of that appreciation, along with these great mentors that I had at the time, that ultimately steered me into a path of research. And I ended up doing classical so-called wet lab laboratory research, which is when you go into a lab, put on your lab coat, play around with cells and DNA molecules and protein molecules, and really understand the kind of nuts and bolts of how cancer cells work. And very interestingly, through that process, probably after 3 or 4 years during my PhD, that was my essentially PhD period, which I did at the Walter and Eliza Hall Institute here in Melbourne, started to get very interested in an aspect of cancer biology that was appreciated to some degree prior to that, but in that era it was a very hot, interesting topic in cancer research, which actually directly relates to the problem clinically that I saw in those years of training of patients becoming resistant to treatment. And that is how cancers, in many cases, have this intrinsic adaptable nature that enables them to change and evolve, even in the face of receiving treatment that's supposed to kill those cells, and ultimately enables the cancer as a whole to become resistant to the treatment. So the sort of molecular mechanisms, if you like, through which that adaptability could develop, I became very interested in the latter part of my research. I think particularly because it, to me, relates to the number one clinical challenge in cancer that we had then and really still have now. And as I've just articulated, the fact that in essence cancers are in many cases almost like living organisms. the human, I mean, we've evolved as human species fundamentally because of the ability of living organisms to adapt to their environment. That's how we've sort of ended up how we are, and we're still evolving, and we'll keep evolving going into the future. Of course, human or species evolution happens over, thousands, millions of years. But in fact, the same principles actually hold for to explain the mechanisms through which cancers, which you can consider in this context as also as living organisms, albeit living inside someone, also have those same types of properties. So where the individual cells have got the ability to adapt and survive in the face of some sort of attack by a person's immune system, or maybe by a toxic cancer drug that the patient's being treated with. Although the cells come under pressure and they stagger a bit, and some of them die and get killed, which is why our treatments do work. But in many cases, not all the cells are eliminated and get killed. There are some cells that adapt, survive, change their fundamental makeup, and to enable them to be sort of permanently resistant to whatever that treatment is. And then those cells are the seeds then that grow the new and recurrent cancers. The aspect of cancer that confers this ability is kind of collectively known as cancer heterogeneity. The idea that although a cancer is like a lump of cells, within the cancer, many of those cells are actually quite different from one another. So they all arise originally from the same, if you like, cell of origin, the same source cell, that then becomes malignant, cancerous, multiplies, divides, spreads. But in the process of that division, they actually, usually in small ways initially, end up becoming different from one another. And some of them just probably randomly acquire the ability to resist treatment. And so they're the ones that get left behind after a period of treatment. They sort of kind of bunker down, survive, but then when the treatment pressure comes off, or even sometimes during ongoing treatment, they start to get active again and divide and multiply, and they're the ones that form the recurrent or resistant cancers that actually ultimately kill. I mean, that's basically the reason why why the cancers that actually kill people, that's actually how it happens, is because of this fundamental adaptive nature of the disease that enables it to resist treatment.
So they sort of go dormant and come back, isn't it?
Yes. There's evidence that some mechanisms of resistance to treatment is via an initial period of so-called dormancy, where the cells just bunker down. That's right, they just sort of stop doing anything. They don't divide, they just sort of sit there, but they're not dead. And so when that treatment pressure comes off, then they can get active and start to multiply again. But the problem then is that they're already, because you've sort of got rid of the cells that were sensitive to the treatment, because they're probably gone, the problem is that the recurrent cancer that you have has this to some degree, intrinsic resistance to the treatment that you've already had. And that creates increasing challenges because we just run out of treatments. So the phenomenon of cancer heterogeneity towards the end of my PhD was something I became super excited about, interested in. I still am. I mean, to me, it's still, I think, the biggest challenge that we have in making quantum advances in cancer treatment going into the future. The reason why cancer immunotherapy, I think one of the main reasons why cancer immunotherapy has been so remarkably and surprisingly effective is because cancer therapy doesn't actually affect the cancer directly itself. All it does is to activate a patient's immune systems to respond to and adapt and actually kill the cancer. And I think one of the reasons why it's been so effective is that initiating an immune response essentially triggers a very adaptable response. So it's like as the cancer is adapting in the face of the pressure of being attacked by these immune cells, the immune system actually kind of automatically adapts at the same time. So the power of our immune systems is that they're intrinsically very adaptive. So sort of change what they attack and when they attack based on the whatever signals that they get to start that attack. So whereas if I just give someone like a chemotherapy drug, it's just a molecule. It doesn't have any intrinsic ability to change. It is what it is, and it'll target that particular chink in the armor in a cancer cell, and hopefully the cancer cell dies. But if there are some cells that are resistant to that treatment, then the chemotherapy is no good anymore, because that drug can't change itself to adapt to the new state of the cancer cell, but the immune system can. So the immune system can actually adapt to new signals and new sort of signs of danger in the body, if you like. The immune system's a remarkable and remarkably powerful system in our bodies. And the reason why immunotherapy has been effective is because to a degree it overcomes this intrinsic adaptability and ability to acquire resistance that cancers have to many treatments. In immunotherapy treatment, even as the cancer is trying to adapt and become resistant to the immune attack, the immune system is like immediately adapting to match it. It's a bit like playing defense in basketball. You've got a person coming at you that is, sort of going from side to side with the ball, but the defender keeps sort of moving to keep the attacker in check. So it's that sort of concept. Whereas the concept of a chemotherapy drug is like, it's just like a basketball defender just standing still. And sure, you can't get past that. If you're a shooter, you can't get past that person. But if you go around them, you can if they just stand there. But the really good defenders are the ones who move and adapt, so wherever the attacker comes from, they can keep them away. So I think immunotherapy is a bit more like that. Whereas our sort of old-fashioned drugs, if you like, chemotherapy, they're more those sort of static defenders.
That's a great analogy. How do you get them to move?
How do
you get the defenders to move?
Well, we're lucky because our immune systems intrinsically are mobile, and they can change and adapt. So that's just how they're built, with legs that help them run around and adapt, basically. So that's not a great metaphor, but yeah, it's just the intrinsic nature of the design of the immune system and just how it's evolved. If it didn't have that adaptability, then we'd all be wiped out with the next minor change in the flu virus that our species wouldn't be able to cope with. But it's that sort of ability for the immune system to rapidly adapt to changes in viruses and bacteria and things like that we routinely encounter all the time. Our immune system's constantly changing and adapting to deal with those new pressures and those new dangers. It's the same. So they're basically evolved like that. That's why we're here. And immunotherapy harnesses that adaptive power.
Could you do me a favour and give me a brief timeline of immunotherapy from how recent is it? When did it start? And it's come a long way in a short time.
The story of immunotherapy itself is absolutely fascinating, and actually has got a lot of lessons beyond just how to develop new medical treatments. Certainly new cancer treatments. So, the first reports of immunotherapy being active against cancer, like at least some type of immunotherapy, actually go back like 150 years nearly. So there was the first sort of published immunotherapy was something called Coley's toxin, which was essentially just basically a bacterial, essentially a poisonous bacterial product that was injected around cancers in the skin and induced very strong immune responses, because it was like a nasty bug that was being injected around the cancer. But fascinatingly, there are reports of cancers around the sites of injection actually shrinking down just with that injection, presumably as a result of the immune system being activated primarily against the bug, but because of all the sort of generic activation and inflammation that was occurred, there was some essentially probably some what we call bystander immune effects that resulted in the immune system not only getting rid of the bugs in that case, but also attacking the nearby cancer cell. But look, the truth is not a lot happened thereafter for like 100 years. And it's actually one of the great stories of medical research perseverance, that we have the immunotherapies that we have today. So a lot of the advances in understanding in immunology kind of date back to the 60s. So these are really fundamental understandings. I mean, and in fact, Australian researchers contributed very substantially to that through the 20th century, actually. But a lot of the really detailed understanding of the immune system was, enabled, to a degree by technological breakthroughs through the 60s and particularly 70s. And so those initial kind of discoveries and learnings pointed some cancer researchers, but only some, in fact a very small number, to the possibility that this new understanding of how our immune systems work could be kind of harnessed to educate immune systems to attack cancers. And so I was actually quite fortunate in that period of the sort of latter part of my training when I became interested in cancer research to be working actually in a group here in Melbourne that was really very actively doing cancer immunotherapy research by trying to essentially develop treatments conceptually similar to injecting nasty bugs. They were essentially trying to induce immune responses, so to give the body a patient's body some sort of warning signal that the cancer was a bad thing that it needed to get in and eliminate. I wrote clinical trial protocols. I injected, all sorts of things into patients to try and induce. They were all done via clinical trials. To try and incite or induce immune responses in those patients against their cancer. But interestingly, that approach of trying to induce immune responses, apart from some occasional surprising and often spectacular successes, they were very few and far between. So sort of most clinical trials back in those days would enroll maybe 10 to 20 patients, and these were patients that had basically no other options. They'd exhausted everything. And maybe you'd get like one patient who responded out of 10 or 20. So it's like, there might be something there because it seems like there's an effect, but you can't really do much with like a 1 in 10 or a 1 in 20 response. There's sort of nowhere to go with that. So it seemed there were some clues that this was the right thing to do, but it wasn't really clear. And I can tell you that, actually literally working in that field with some of the leading people in the world at that time doing that actually these guys were pretty outside of the mainstream thinking. This is only like the mid to late 1990s, not that long ago. It was really hard for those groups to get competitive funding from NHMRC and other sort of international funding through international other funding mechanisms. No one really believed it. At the end of the day, the mainstream thinking was that it just can't work. Like, it's just not going to work. And all you guys do are trials of 10 or 20 patients, and one patient responds, so what? This is not going anywhere. So it's actually a story that's really needs to be told, because there were some, if you like, true believers who couldn't put down the possibility that this would work in some way, and if it did, it would be incredibly impactful and influential. And there are some key figures, I think, that were very influential at the time, particularly a guy called Lloyd Old, who was the director of an institute called the Ludwig Institute, a philanthropic institute based in New York that had branches all across the world, including 2 here in Melbourne at that time. And essentially Lloyd and a large group of really amazing scientists that were supported by that group. Well, these were some of the only leading lights in the world doing work in this space. And in essence, they were largely funded internally because of the philanthropic support of the Ludwig Institute. And look, there were others outside of that institute that were also important, and I'm not saying it was all them, but the point I'm trying to make is that there really weren't many people that really thought that this was a thing and that this was worth doing. But they did, they believed very strongly what they were doing. They were very persistent. They did really good science. So they were the kind of method that they took was careful, scientifically correct. They produced data that was reproducible and believable, even though it wasn't that exciting in terms of in terms of its efficacy. So sort of gradually, incrementally, the field was advanced. And so this is like the late 90s now. So at the same time, maybe a few years earlier, the other major breakthrough was, so sort of prior to that era, most of the understanding of how immune responses work was focused on how you go from having no immune response to some immune response. So how's the response activated? And then there sort of came a latter period of research, again, sort of late 80s, early 90s, which was focused on something that you wouldn't really sort of think might be important, but it turns out it's super important. And that is, if you get like a cold or something and you feel crook and you feel awful and you lie down, and because your immune system is going haywire inside you, getting rid of that virus, the virus does go and you get better. And that happens, and you start to feel better. And that happens substantially because that immune response is actually shut down. So the immune response doesn't... So if you get a cold and you feel awful with fevers and chills, which is the immune response, you don't feel like that for the rest of your life. Although, I mean, if you did, you wouldn't survive, actually. The immune response gets shut down. And so the kind of fundamental biology of how that happens, of how immune responses are actively inactivated, to just go back to the normal steady state. A lot of that biology was sort of cracked open sort of secondarily in that sort of latter 80s and early 1990s period. And so some of the key molecules that are present on immune cells that really regulate that sort of active shutting down of an immune response when it's not needed anymore. A lot of that was only just being understood and at a very fundamental level at that time. But of course, then these clever cancer immunotherapy researchers, coming in a few years later, were looking at this field and going, okay, well, that's interesting. So what if we actually in cancer patients, because we have some evidence that their immune systems are already active against the cancer. There's a lot of evidence that. What if we try and stop their immune responses, so actively try and stop those immune responses being shut down? So kind of keep the immune response active. And wow, that was the key. That was the trick. So if you get in the way of mechanisms that actively shut down immune responses, which we normally have in response to bugs and viral infections, and which very surprisingly cancer cells hijack incredibly well. If you shut down those mechanisms that themselves shut down immune responses and keep the immune system responsive and active, which is essentially the basis of the modern immunotherapy drugs, then you kind of release the immune system to be an incredibly powerful tool against cancers. So it turns out that after all this sort of years of discovery, that these guys essentially figured out that that's really the key chink in the armor. It's not so much that our immune systems don't respond to the presence of cancers. Like in a pretty high proportion of cases, maybe 60%, we think at least, maybe more. There is, the immune system. Our immune systems recognize a cancer inside us as being a bad thing, and make an initial response to try and get rid of it. But in a brilliant sort of moment again of adaptations, we talked before about cancer cells being able to change and adapt. It's like in a kind of brilliant, rapid, real-time evolutionary moment, cancers start actually producing proteins that actively shut down the immune response. Even as those cells are charging into the cancer even as the immune cells are charging into the cancer to do their thing and release the toxic chemicals that kill the cells, the cancer is sort of immediately kind of adapting and sending out protein signals, protein-based signals, that basically shut down the activity of those cells. It's a remarkable bit of super clever adaptive biology that goes on. Cracking that open, and a lot of that work was actually done by this same group of researchers linked to the Ludwig Institute back in New York. So that's why I'm making this point about how important that particular era was. 10 years later, we have drugs that are being given to patients inducing spectacular immune responses and durable remissions, probably cures, in patients with cancers that hitherto would have been fatal within 2 or 3 months. So it's an extraordinary story, and it really highlights, I think, from a societal research, medical research funding point of view. I mean, if you think about it, I mean, if that sort of group of researchers in that period hadn't been able to kind of cobble together funding and get a supportive philanthropist actually, or work within that sort of context, we wouldn't have had those advances, and who knows where we'd be. Probably advanced in some other way, but it just highlights that. At the time, the mainstream. So now, like, the mainstream is, of course, immunotherapy. Yeah. I mean, it's obvious, it's an obvious thing to do. But I can tell you, back then, this was like, I used to go to meetings, conferences, and because I was working in an immunotherapy trials group, I used to go to the immunotherapy talks at the conference and see what people were doing from different parts of the world. There was only a dozen people in the room. I mean, this was a conference of 20,000 people, and there was almost no one going to the immunotherapy sessions because those guys are all crazy, so no one went there.
Not sexy enough either.
Well, yeah. So, but it just highlights the fact that you don't always know where the next big idea or breakthrough is going to come from. So I often get asked the question, like, what's the next big thing? And my answer to that is, whatever I'm going to say, as a member of the mainstream, I guess these days, it's like a 99% chance that I'm completely wrong. So you can essentially ignore everything I'm going to say, because I think it's true. I think it's actually really hard to predict where the next big breakthrough is going to be. I think the key point, particularly for research funding, is that it's really important to sustain a diversity of ideas and techniques, and I think technological innovation is key. That's often a driver of new areas, but being able to sustain a diversity of ideas across in this case, medical research, across the medical research sector. So not sort of throwing all of your bucks to one idea, but I mean, you still need to support preferentially the stuff that's sort of looking good. There's no problem, but you can't forget about the kind of other, maybe slightly left of field ideas that are out there that will otherwise wither and die if there's no support, because it might be that the next big thing is actually hiding in some little conference room session with only 12 people there that no one really cares about or sort of go to, but that might be where the next big thing comes from. It's actually a bit like evolution itself. So that's kind of what evolution, fundamentally evolution and actually cancer treatment resistance that I talked about before, essentially relies on that diversity within a population. So you've got to have diversity within a population so that whatever, from an evolutionary point of view, for the species to survive. Because whatever sort of comes along, you want to make sure that you've got at least some one cell or one person or one idea which is well primed to take advantage of that new environment. It's actually a similar kind of concept.
Stu?
Yes.
We've got I think there might be someone else who needs to use the room. We've got another minute or so, I think. Is that okay with you, Mark?
Oh, God, that was fast.
Yeah.
I've obviously talked to you. I told you I'd ramble.
So it would be good to.
I can double check if you guys need more time.
I can. I mean, if I'm a bit late to this, I'm not sharing the meeting, so it's okay. I can be a little bit.
Well, just in the ACRF grant,
Speedy.
I think, if I understand it right, that gave you some equipment that enables you to look at things, whether the immunotherapy is working in real time. So I guess there's an interesting sort of area, the role of tools in actually knowing how you're going, because it seems to be research is about brains and tools, So if you could talk about that
Extrapolate
somehow, yeah. And mention ACR.
Yes, of course, yeah.
But not by initials, by my name.
Yeah, so there's no question that quantum advances in medical research and our understanding of biology in the world are often driven by the availability of technologies, and particularly new technologies. So as much as it's important to support a diversity of ideas across the research sector, it's also super important to support technological innovation. So tools, the development of tools to be able to do things and ask research questions that you couldn't ask if you didn't have those tools. I mean, you can't undo a Phillips head screw if you don't have a Phillips head screwdriver. You know what I mean? So you've got to be able to. So having new and innovative tools enables you to do things that you can't otherwise do in research. So, research funding schemes such as supported by the Australian Cancer Research Foundation that are really focused actually on basically on equipment innovation, on supporting the development and application of new research tools better to understand cancer. I think these schemes are really, really critical, and they work very nicely side by side with other types of funding schemes that support people or project and research ideas. One of the things that we're really focused on is trying to now study, because of the availability of new tools, particularly very advanced PET scanning technology that's increasingly being installed in locations across the world, not many, but some, and here coming soon, is the ability to study immune responses that occur in patients because they've been treated with immunotherapy drugs in real time. So most of our understanding of how the immune system responds to these immunotherapy drugs, a lot of it's actually based on studies in non-human models, actually, in other animal models in terms of the initial research. Now, we do understand a fair bit in terms of the way that patients respond to immunotherapies, but there's a lot we don't know. One of the challenges has been that how do you actually sample or how do you actually follow and test an immune response in a patient in real time? Traditionally, we've done that by taking like the occasional blood test or the occasional blood sample after a patient's had treatment. That might be taken, I don't know, 6 hours after treatment or a couple of days or a week. But at the end of the day, it's like a sort of stochastic, and very minimal sampling, actually, if you think about it. I mean, you're only sampling blood, and you're only doing it at a relatively small number of time points. So although that's a form of real-time monitoring of the immune system's response to immunotherapy, in fact, it's very, very minimal. The beauty and the power of quadrupet that we'll be applying in this research program is that with this technology, the resolution with which we'll be able to study immune responses in patients treated with immunotherapy is off the charts compared to what we've previously been able to do. And we can literally sit there on the scans and watch even individual cells or subtypes of immune cells track not just within the bloodstream, because many of the immune cells end up out of the bloodstream and into tissues. We can see all that via this high sensitivity total body PET technology. And we are very confident that this will provide us insights into how immunotherapies work, into what types of cells are even activated in the body in response to immunotherapy. And we really hope give us a much better ability to predict which patients are going to get sick as a consequence of immunotherapy, because these drugs do have side effects and toxicities, and they can make people feel unwell. We've got pretty good antidote treatments, and we can usually get on top of them well. But the sooner we know that those problems are arising, the sooner we can hop in with those sort of antidote treatments and prevent or minimize people getting sick from the treatment, then the much better long-term responses that we're going to see.
Great. I'm pushing for time. Could you summarise what the study is in a very quick summary of what you're doing, of the new?
Yeah, so the ACRF Centre for Dynamic Immuno-Oncology aims to study in real time patients' immune system responses after they've been treated with immunotherapy for their cancer, to understand the nature of those responses, where immune cells travel around the body, and really which patients are likely to respond to treatment based on those initial immune responses, and also which patients are going to get side effects and what the nature of those side effects is, so that we can intervene with treatments to prevent the side effects getting really serious.
Have you had many eureka moments
I've
in
been,
your life?
yeah, I've been, apart from having kids and stuff, and from a work point of view, look, I've been super lucky in my research career to work in some great environments with some amazing people. I think there are probably 2 eureka moments that really stand out. The very, very first part of my PhD, actually before I got interested in cancer heterogeneity, I was actually just studying normal breast biology, at least as it pertains to the development of breast cancer. And our group and me with my research were basically the first people in the world to grow a fully formed and functional breast capable of producing milk from a single breast stem cell. This was a huge innovation at the time. This is like 20 years ago now. But the time I looked down the microscope and saw this fully grown in the laboratory system that we were using, and saw this fully grown breast after I just literally injected one cell into that model, was a mind-boggling moment. And I remember the whole lab was just going crazy, looking down the microscope at what we saw. The second moment was about 5 years later, during what's called my postdoc training at the University of Michigan in the US, where we did a sort of conceptually sort of similar thing, injecting single human cancer cells, in this case melanoma cells, and being able to grow remarkably robustly fully grown melanomas, again from the injection of single cells, which really had very profound implications for the nature of melanoma cells, indicating that they are there aren't many single cells in a normal body that can survive the rigor that we put these cells through in this laboratory experiment, and that single cell tumor forming experiment. The first time we saw it, we just couldn't believe it, that this was even possible, because no one had ever done anything like that before. And what it really told us was, just how difficult these cells are going to be just how difficult it's going to be to kill these cells, because they have a remarkable, this sort of idea of them kind of bunkering down and adapting to very stressful situations. That's what these individual single cells were doing. Now that was of course before the immunotherapy, which came about 5 years after that, and which has changed some of the experiments that we do. But yeah, the ability to grow very complex biological systems, if you like, so a complex mammary system capable of producing milk, and a complex human cancer environment or tumor from the injection of single cells, they were 2 like remarkable moments in my career.
We better do one more question about, how we ended up with you downstairs.
Yeah.
It's got to have some context about where we are now.
Of course.
This wonderful building and everything that's been achieved here, and the philosophy behind it, if you wouldn't. And quick, because we know you've got phone calls.
Of course. Yeah, so I've been very fortunate to be in the inner sanctum of conceiving, finding funding for, and developing the Paula Fox Melanoma and Cancer Centre, which is, to a degree unique in several ways. The first way, I think, is just its design, and it's very patient-friendly focus, and that's all kind of purpose-built architect design. It's a place that doesn't really feel like a hospital when you come in the door, and even when you're having treatment. I mean, there's nurses and doctors around. It is a hospital, but it doesn't really feel like that to many patients. And I've had lots of feedback from patients exactly along that line. I think the other really key feature about it is that this is not a standalone entity. So we're basically we're just another wing of the Alfred Hospital, from a governance point of view, from an operational point of view. So although this is a special place and it's a bit different from the regular hospital, and the patients love that aspect of it, we have access to the full range of really top level medical treatment resources that are here at the Alfred. So it's like, although you come into this building and we're focused on your cancer treatment and you're in a nice environment and it's very patient friendly, it doesn't matter what happens to you under this roof. You've got the full support of one of the world's great hospitals right at your back.
I've got two more questions actually. How are we for time? Pushing a friendship? That's the one.
That's okay. Well, as long as Mark's happy, then that's okay.
Yeah, I'm not chairing the meeting, so it's not.
You spoke of research for research's sake, and the unexpected results of research, I guess, and you and I had the conversation in Sydney. It's like, well, without the space program, wouldn't have had the microwave. You never know which way research is going to go. And if you could just take that thought and compress it a bit, I'm compressing your answer for you.
Yeah, so I guess to jump back to that theme about, it's really important for our society and the environments we build to support research that a diversity of research ideas is maintained. We can't throw all our eggs at one idea. And I do get a little bit worried these days about the amazing amounts of money, I would say almost ridiculous amounts of money, that are being thrown at new technologies such as AI. I'm not saying AI is a bad thing to invest in. It's actually amazing. But we've got to be really careful to throw all our eggs in one basket, because it's really hard to predict the future. And there might be a pocket of cancer immunotherapy like researchers hidden somewhere, not able to get funding, not able to do the research that they want to do because all the money is going to AI research, for example, that actually those people are actually the group of people that really will foster in the next major breakthrough in research. So we need to be conscious of sustaining that sort of diverse ecosystem of research ideas. It's critically important.
And what do you love about your job?
I keep saying diversity, but I think actually it's one of the things that I really like about my job. I mean, I'm very privileged to work in research, and that certainly gives me a lot of energy and ways to, I guess, develop avail my kind of creative juices, I suppose, because research at its core is actually very creative. But I actually love the fact that I still work as a doctor. I still treat patients. I'm very involved in the sort of overall running of the hospital and working out efficient and patient-friendly ways to deliver the best possible cancer services that we can, usually within a pretty limited budget. And I also, particularly in recent years, have really enjoyed the opportunity to think about the equity of access to healthcare in our system. Even in Australia, we have an amazing healthcare system, but there's still incredible inequity in the ways that people across our community can access that healthcare system. So it's been a real privilege, even just the last 5 or 6 years, to be involved in programs of regional outreach and essentially restructuring the way that we deliver care, to make sure that people, as much as possible, get the best treatment they can, regardless of their postcode. I think it's such a critical point.
I'm going to go, but I'm just going to check with Ben, unless he's got his hand up.
Yeah, well, what you said just then about diversity, the heterogeneity of cancer, it sounds like we need to be like cancer to combat cancer. That we can't put cancer would never put all its eggs in one basket. it's going to be doing this, that, and the other. So we're going to have to.
Good, just to make it-
Completely agree. Yeah, I completely agree. I think this.
You're going to have to say what you agree with.
We don't hear the question.
Of course,
No,
yeah,
exactly.
yeah. Yeah, so I think that, like it's essential for the survival of our species it's essential for there to be diversity across our population in terms of different types of people. It's also important for cancers to have at least cellular diversity across them so that they can resist treatment. It's also critically important in supporting a research ecosystem that is sustainable and able to generate the next big breakthroughs, that there's maintenance and support of a diversity of research ideas across our community.
You can clap. I'll clap.
It's
I know you're stuck.
yeah. I actually think it's a great analogy, but it's not an easy one to it's not an easy one to say very simply, I think, because it's obviously what cancer and research and funding and
Yeah.
what's that mean. So
I think it's easier to say if you don't know what you're talking about, because it's not complicated by all the knowledge.
yeah,
But
right.
It must. I mean, I'm intrigued as to why immunotherapy wasn't adopted earlier, because it
Well, I mean, so one argument that's a flow on from the comment about diversity is, one of the reasons that immunotherapy didn't become a thing until relatively recently is because no one really believed in it back then. And in fact, back in that era, most of the money and funding and research was being directed towards actually genomics. That was around the kind of birth of the high throughput genomics era, sequencing of the human genome occurred in the 1990s. So there was so much money being poured into that area that there was no money going into immunology, into cancer immunology research, and very little into immunotherapy research. And that's my point. I mean, if you kind of spend all your bickies on one idea, even though it might be a good idea and there might be something in it, but if it's at the expense of a lot of other ideas, then the potential is there to slow progress, particularly if those other ideas contain the next big breakthrough.
That was well said. Look, I'm happy and you're late.
brilliant.
Okay.
Yep.
Diabetic.
Here we are in the nerve centre of the Paula Fox building. This is level 2. It's the location where patients come to receive cancer treatment, usually chemotherapy, but increasingly these days some of our newer treatments, such as immunotherapy. It's a really purpose-designed facility that is designed in the way that it is to try and make patients feel as comfortable as possible, and also to make it feel as little like a hospital as possible. And that was done really with deliberate design, particularly by incorporating a lot of features, so soft forms such as wood, and a lot of curves around the spaces that patients occupy. And this has the effect of making them feel like they're at home.
That was great.
Here we are in the nerve centre of the Paula Fox building. This is the level 2 facility where patients come to get treatment for their cancers, medicines such as chemotherapy, but increasingly some of our newer medicines such as immunotherapy. This facility has really been purpose designed to make it feel as little like a hospital as possible. A lot of the architectural features are very purpose built, a lot of wooden panels, a lot of curve and sort of comfortable spaces that make patients feel as much as possible like they're at home. Here we are in the nerve centre of the Paula Fox building. This is the level 2 facility where patients come to get treatments for their cancer. Often medicines such as chemotherapy, but increasingly in the modern era, immunotherapies that give great... so I've just lost that train.
Should we start again? Can you start right?
Start walking again, or?
Yeah, I think so. Maybe we'll just have it just there near the stage. It's all right.
Walk in once more, I think.
All right, very good.
Sorry. No, just come through. Just do your stuff. Here we are in the nerve centre of the Paula Fox building. This is the level 2 facility where patients come to get treatment for their cancer. Medicines such as chemotherapies, but increasingly other options such as cancer immunotherapies that can be very effective. The facility's really been designed to make it feel as little like a hospital as possible. With the wood panelling and the curved spaces, these really make patients feel as much as possible like they're at home.
And is there a benefit to that?
We know that we've had a lot of feedback in the process of developing the building from patients and consumers that it was really important for them that they didn't feel like that they were in hospital as much as that was possible. It's hard enough going through cancer treatment, let alone feeling like you're in a very, very foreign environment. And that was the feedback that we got very strongly.
Yeah, go ahead.
Grant?
I don't
Grant? Thank you.
Do both. Okay, so.
It's
Okay, roll on.
legible, okay.
Roll. Ring the bell 3 times well. And just ring it again once. Should we?
Yeah, I'm coming. I did
And
get.
go. Just
I
ring
did
it
feel the
once and then
motivation,
out.
Steve.
And ring. And out.
Sorry, one more for me. Sorry. Chatting.
And just hold the rope steady. And again. And ring. Okay, we'll do it again and we'll get you to after you've rung it
To clear shot.
Thank you. And ring.
Right, come in.
We'll do another one of those. Up there.
They walk away along the same line, so.
No, I'd walk out. Come across. Yeah. Okay, and rolling up. And action.
Shut up.
And action.